Okay.
Yeah. Can you now maybe? Yes, it's good. So we have Elena and Ilya. Elena is biologist, synthetic biologist from Paris. I mean, she's French. She's studying now a PhD in Delft, Holland, and she's actually studying ways of communicating synthetic biology outside of the academic world. And we have Ilya, who is also a biologist. He is founder of London Biohack Space. If you go to London, check him out. It's actually here, the address and Twitter and stuff, and we're going to talk about genetic engineering in the next hobby. So welcome, Ilya.
Okay. Okay, so I'm not a founder, I'm actually just one of the directors of London Biohack Space, just say that. But
yes.
so the idea here is I want to talk to you guys about and maybe enthuse you that maybe this is something you should get involved with if you know nothing about it so far. This is something that maybe if you're interested in the maker movement, you might be interested in making with biology, I guess. And that would be genetic engineering, or some people call it synthetic biology, or genetic modification, or whatever you want to call it. And so how can you do genetic engineering outside of university or outside of industry, right? How can you do it as a hobby? Because that's kind of what I do. And so the London Biohack Space is kind of a subgroup, a division of the London Hackspace. So if you go to London, visit the Hackspace, this is what it looks like upstairs. Just a very big Hackspace of over 1,000 members. And we've got a nice big space upstairs with 3D printing, laser cutter, electronics, everything like that. And then in the basement we have all sorts of tools and woodworking, metalworking, lathes, robotics, whatever. But the cool part is that we have this. So this is actually from a few months ago, so since then the lab looks even again different. And so this is the biolab. And this is all the stuff that you basically need to do some basic genetic engineering. So we're not kind of competing with universities, we're not doing crazy out there projects. It's still, I guess, getting to grips with things and getting things working well, because we're doing things cheaply. And so that means sometimes you have to accept that it doesn't always work. And with biology, things are often a bit slower. There's a lot less kind of tactile feedback than there might be with carpentry or with electronics or things like that. There's a much slower iteration time. So when you fail for longer. So you have to plan even more, right? But anyway, so the point is this is a working laboratory, right? Even if it's not 100% perfect. Here's just another view of it. And so just to give a quick explanation of what a lot of genetic engineering or synthetic biology essentially boils down to is we take some DNA from somewhere, from some organism. We have a gene that we're interested in. We might be looking for a specific set of genes, and we find out what we want. And from there, we look in databases and things like that. And there's lots of resources around that. And we get the sequence for that gene. And either we do that, we biologically copy it, or we do it digitally, and then we can pay for someone in a company to synthesize it and then just post it to you. And that's now becoming cheaper and cheaper. And we put it inside what's called a plasmid, which is just a little loop of DNA. And so we insert it with some wet lab experiments, which is essentially using enzymes, or various others, a whole bunch of different methods for doing this. Some cheaper, easier, some more expensive, harder, whatever, a whole bunch. I'm not going to go into the real nitty-gritty detail of it all, because we'll be here for hours otherwise. But so essentially you put it inside, you insert it into that loop of DNA, and you use one of a couple of methods of putting it then inside the bacterium. And the nice thing is, if you're working with bacteria, so often we work with E. coli because it's very well characterized, a lot of is known about E. coli, and so it's kind of, for that reason, much simpler to work with, and it grows quickly, a whole host of reasons it's great. And we put it inside, so I'll talk maybe a little bit about that, but and then grow it up and grow lots of them. And now hopefully whatever gene you put in there is going to be doing what you hoped it to do. So this might be a gene that expresses a particular protein that you want to do, or several proteins that form a pathway that perhaps let you synthesize some compound you're interested in. right? And at that point, you have a bunch of bacteria doing what hopefully you want them to be doing, and you can make them multiply and grow them up. And that's the basis of genetic engineering, right? And so I'm going to talk a little about practicalities of getting to that stage of where you can start doing these things in a hack space. And so it's great to have all that equipment and that, but one of the most important things, certainly in the UK, is to essentially you need to have a license from the central authority to be able to work with genetically modified organisms. And so we're the first community lab in the UK that is allowed to do this. So we have CL1 status, and it was a lot of kind of reading all the legislation and things like that, and essentially drafting standard operating procedures that encompass the fact that we're a community lab and that the government is essentially happy with. And so we put all our documentation up on GitHub, and the hope is that other places in the UK, and I guess the rest of the EU has reasonably similar laws. So it should be applicable, so you guys, it's all there if you want to kind of start doing this stuff. For you to kind of tweak it and apply it to, you obviously have to look at the laws in your specific countries and see how it applies, but the framework hopefully is there. And so then, you end up with a letter that looks a bit like this, and then you're basically good to go, right? Except obviously you need a lot of this equipment. So how do you get all this equipment, right?
And mate.
And good mate, right? That's important. So we get, so one of the nice things about working in a hack space, right? I'd say the important thing to start with is you need a group of people who are interested in doing this. So, how do you find those people? That's question one. Question two is, where do you get the equipment from? So, in a hack space, and where do you get the space from, even before you get the equipment? Where do you get the space from? Right? In a hack space, you have those first two things. Hopefully, there might be some people, other people like this, hackerspace. You kind of post on the mailing list, whatever. You say, hey, who's interested in doing this? Who's interested in exploring this with me? And get together and see what happens. And secondly, if you're in a hackerspace, maybe they'll be willing to, devote a little corner, or maybe not, just temporarily, some space and some resources to being able to work there on these sorts of things, right? Eventually, if you want to do the GM stuff, then you're going to need a dedicated space. But maybe that can come later. And then you're also going to need some knowledge, right? Because one of the barriers to all this stuff is there's quite a lot of knowledge to kind of intake before you can even start to appreciate like how to start doing these things. So this is kind of more a taster than what do you do, like how do I get going? But one of the handy things is, so there's a bunch of resources here. So Open Lab Blueprint is made by GenSpace in New York. I think it can be improved, actually I have plans for kind of maybe a better thing, but it's basically a list of equipment that you need to do a whole bunch of this stuff. And where to source it from, maybe, right? Or where to build it, how to build it. So there's a lot of open biohardware being developed, and that's, some of it is reliable, some of it not so. And one of the nice things about being a group with a dedicated space is you can find your local university and say, hey, I see you're throwing out some equipment, can we have it for free, please? And in our case, they often say yes. And the great thing about old biohardware from so all our lab stuff is kind of mid-90s, mid-2000s era, right? And it's fine because these days, genetic engineering, the thing that's improved and the thing that's changed is the chemicals you use in the equipment. But a centrifuge is still the same centrifuge as people used in the 80s, right? An incubator is just a warm box. PCR machine, again, it's the same thing that was being used in the mid-90s. Nowadays they have touchscreens, but you don't need one. There's a whole bunch of stuff. So this old equipment works great, and it's because it's proper lab spec, it's been manufactured to specific tolerances, and it's in many ways, I would say, a better route to a lab than trying to build your own hardware, because you're going to sink a lot of time figuring out how to do the electronics and stuff like that, and it might not work so great in the first place. Whereas if you end up with a whole bunch of stuff from a university, either it's slightly broken and it's much easier to fix it than build a whole new one, or it works in the first place, and that's great. And then, so then there's the DIY bio mailing list, like that's an excellent resource for talking to people who are interested in this stuff from all around the world. And OpenStax College, I think when you need when you want to get into it, you need all this knowledge, right? And I think at this point the best thing to do is to pick up a textbook. Genuinely, like just pretend you're in college, right? And this is an open access set of textbooks, and they have a thing called Concepts of Biology, or just Biology is another one. And it's open access, it's actually quite good. And there's also synbio.info, which is kind of like a wiki style, what you need to get going with synthetic biology. Okay, so how does all this equipment kind of go? This is a kind of blueprint of our lab, I guess. So we've got a -20-degree freezer, some centrifuges, incubators, PCR machine, autoclave, and a fridge, and a bioreactor, right? Bioreactor, I'd say, is optional, and a laminar flow head. So what do we do when we do genetic engineering? So first things first, we need to sterilize some of the kind of nutrient soup that we're going to be growing our bacteria in. And so you need something like this, which is an autoclave. If you're going to be doing GM stuff, you also need it up to spec so that you're able to sterilize things that are genetically modified before you throw them into the waste, right? So that's why it needs to be up to spec. If you're not doing genetic modified, you can get away with just using a pressure cooker. So that's the cheap alternative. Whereas our one we bought for £50 on eBay. So there's normally like around £1,500 second hand, but because someone didn't know how to fix it and it was a very easy thing to fix, it was £50. So that's cool.
You can use a pressure cooker.
Yeah, you can use a pressure cooker, but you can't use it to dispose of GM waste, right? So we had to have something like that. Anyway, so this is the electroporator. Again, this was donated. This was like an old prototype from like the mid-90s, you can kind of see. And it was broken or was missing a part, so we kind of made our own. It works great now. And this is what you use to zap the cells with about 1700 volts. And that's what gets the DNA inside them, essentially. Right, and then you've zapped, so you've kind of made your nutrient soup, growing your cells in that. Then you zap them with your DNA, and then you need to plate them out. And this is something that we built ourselves. This is like a laminar flow hood, and what it does is this is a HEPA filter here, and there's a fan in the top, and it blows out a kind of nice laminar flow of sterile air. And what it means is this area in front of it remains nice and sterile. So all the kind of nonsense flying, all the spores and bacteria and everything floating around in the air never touch your samples. So, and that's quite important. I mean, the cheap version of this is if you just get like a camping gas like this, or like a Bunsen burner, then if you work kind of around below it, there's a sort of updraft from the heat that means that you kind of generally avoid getting contamination. But this is a much bigger work area, and much kind of more robust. Oh yeah, and then the bioreactor, so I guess we plate them out, plate out our bacteria, and we put some antibiotics on the plates, and that means that only the ones with our genes inside them are able to grow, and so we've now ended up with just our pure colonies of bacteria that contain the genes we want them to contain. So we grow them up in an incubator, and that, yeah, for ages we were just using a pasty warmer, you know, like just that places had to warm, you know, sausage rolls and things like that. Or you can use like hen egg incubators, or you can basically get a polystyrene box with just a normal filament light bulb in there, and a kind of thermostat as well to keep it at a regular temperature. Yeah, and then if you want to grow it up if you want to get loads of your bacteria, you have to kind of upscale and use something like this, which is a bioreactor. Okay, so what kind of stuff can you do with genetic engineering? So this was like a Hello World thing. We didn't do this. This is a project called Polaroid. Except the thing is, I wouldn't say this is Hello World. This is actually a bit more complicated. So this is more Hello World. This is glowing bacteria. This is GFP. So it's a protein that you get from jellyfish, and it's called green fluorescent protein. And what it means is, when you shine UV light on the fluorescent protein, it glows green. And so here's a flask full of bacteria which is making this green fluorescent protein from the jellyfish. That this is a UV light, and you can see it's glowing nice and green. So that's the kind of Hello World of synthetic biology, genetic engineering, right? What other things? So here's kind of, you know, staying basic, right? Here's some more bacteria that we've put a bunch of coral proteins in these, and so these are proteins, pigments that you normally find in coral growing, you know, in Hawaii or wherever it is. And instead we've got bacteria making them, and so they come out really nice and colourful. So it's kind of like a nice little demonstration. then, so this, I'm now going to mix kind of some of our projects and some other projects that have been done in this sort of area. This is a group called C-Lab. They did banana-scented bacteria. And so here they did the same thing, but instead of green fluorescent protein or coral proteins, they put in a pathway that made this banana scent into the bacteria. And so there's a flask with the bacteria, and if you smell it, it smells of bananas. And then the real vegan cheese thing from Counterculture Labs, and basically they were using yeast that were modified with a bunch of milk proteins. So the idea would be you make kind of synthetic milk using yeast, and then again using yeast to make the enzymes used to turn that milk into cheese. And so it's real cheese, but friendly to animals and vegan friendly. And that was Counterculture Labs. And then this is a project I'm currently still working on called Juicy Print, and the idea here is to make like a 3D printer out of bacteria. And so here we've got, this is just a kind of mock-up demonstration of what it might look like, but we've got this bacteria that grows from kombucha, that grows cellulose in these big pancakes, and the idea is to genetically engineer in a switch in there so you can control the growth of that cellulose with light, and using that to end up making a 3D print. I can talk about that if you need. But, and then this was one of our previous projects for iGEM, the synthetic Symbio Brewery's DIY brew kit. And the idea here was to engineer yeast to do all sorts of different things and make crazy beer. So one that expressed miraculin, and one that tasted vanilla, one that was kind of like red, one that tasted of raspberries, etc.
Can you explain what's iGEM?
iGEM, yeah. So iGEM is basically this big competition, often for undergrads at universities all around the world.
MIT.
And it's kind of hosted by MIT, or it's hosted by iGEM really, but it's held in Boston, a conference about it every year. And everyone comes and presents these projects that they've worked on over the summer. And the concept behind the competition is to come up with some problem and to kind of come up with some solution that uses synthetic biology to kind of solve that problem, or some project involving synthetic biology that kind of is worthwhile in some way, right? So this was our one, and we quite like having fun, right? So we're hobbyists, and so we're not trying to save the world, whereas a lot of the teams there are. And so I just want to finish on this, which was my inspiration for getting into the whole thing, and something I aspire to do at some point in the hackspace is genetically engineer a plant. And this is the human hair cactus. And this is real. This was done in 2001, again by C-Lab. And these are, I mean, it's human hair. I mean, it's definitely pubic hair coming out of quite a phallic green object. So that's what inspired me to do this, because I think the world needs more of this. I think the world needs to have, there needs to be a place to have fun with genetic engineering. I think I'm really inspired by the fact that there's a lot of knowledge and a lot of effort and time required to do these things properly. And I think it's great that there's a space for people to do that for fun, rather than for some monetary end or for saving the world, right? Point is, you can play with this stuff. And I think the more people realize you can play with biology, the less scared people will be of actually important developments, let's say, that use synthetic biology or that use the technology, right? So people play with Arduinos, so let's maybe play with human hair cactuses or something. And so that's maybe my reason for getting involved in hackspaces and that sort of thing. And it's a great way to not only learn, not only if you have a cool idea you want to monetize, maybe that's a cheap way of doing it, but also just to play for fun and have it as a hobby. Cool.
I was wondering about the beer kit thingy. Is that possible to buy online somewhere?
No, so it's not. It doesn't work yet. So it's still being. People are still working on it. Yeah.
It's not good.
I'm a home brewer, so
Yeah, so that was the kind of idea, was to get home brewers involved too. But
So hello everyone, my name is Helena, I'm a scientist, I'm one of the persons, can mention, one of the wet lab persons with the lab codes, etc. So I come from a very traditional academic background. I'm currently trying to become a doctor in the field of synthetic biology. But I'm here to talk not about research and hardcore science, but about something that is really important, is doing science outside the lab and outside the academic work. So synthetic biology is a mess. It's a very broad concept, and even scientists disagree about what it is. If you ask one scientist, you will get 5 different definitions. And the reason why is because too many things are put in the same box. Things like making synthetic cells from scratch, the question of artificial life. Things like using microorganisms to make important chemicals for the industry. Things like making organisms that work with different molecules than the DNA that we have. DNA technology and nanotechnology, biomaterials, genetic circuitry and doing logic gates and computation with biological organisms. And lastly is modifying our own cells to be able to basically engineer the human body. So all of these things are called synthetic biology. And I would say that all of these things have one thing in common, is the application of the engineering principles to classical biology. What does it mean? It means that we consider the living system as something that is programmable. That is the common thing that all of this project has. And so the synthetic biology, what we do is we encode a program within a living system, and most of the time is by programming DNA. And the reason why we want to do that is to be able to control and make them do reliable tasks, which means that you're supposed to make them do a specific task in a controlled manner and predictable, which is kind of like the opposite of what living things do. So yeah, synthetic biology is combining the science of studying biology and the engineering and the practical things of knowing and making. So I put these 2 quotes because these 2 quotes are constantly in synthetic biology talks. So Durand is one of the founders, he's an engineer. He says that testing our understanding by building is the sorter's path to demonstrating what we know and what we don't know. So this is about understanding how light works. And so this is Richard Feynman, a very famous physicist. What I cannot build, I cannot understand. So this is a field that was founded by engineers, not by biologists, which is very intriguing. And the cool thing about biology, synthetic biology, is that for the first time we're conceptually thinking about the fact that we are able to creatively design organisms using rational design. So this is already possible, and it's currently going on in the labs and in research. And so my job is doing that, and you get involved with many things, and you think about what is your job in a more broader context. And so one of those evenings when I had insomnia, I was in the internet and I found the Critical Engineering Manifesto, which I recommend you all. And so the first point about this manifesto is that critical engineer considers engineering to be the most transformative language over time, shaping the way we move, communicate, and think. And that is the role of the critical engineer to study and exploit this language, exposing its influence. And so how do you apply this critical engineering thinking to biological engineering, which is very cutting edge? So this is the thing that I try to explore as a very young and naive scientist. And so because I believe that scientists and the general public, we all have to participate together and engage critically with the complexities of the field. Too many things are important to discuss. So my talk, I'm just going to talk about the different projects that I did to do that and how did it go. So more like telling you a story. So I first started to do some outreach, because to be able to communicate about the field, you need to kind of like explain to the people what it is. So I participated in iGEM this year. We did a 3D bioprinter, so it's fun. So I was super proud of the students because I didn't influence them, but they created a 3D printer that is open source, made out of Next, to be able to print cells in, so it works with bacteria in any kind of 3D structure that you want. So they genetically engineered the cells to produce these nanowires, this kind of like cells, that make the cells stick together very strongly, and you get a very strong biofilm material. And then they came out with this great idea of using Next to make the printer itself. And they were super successful. We won actually the competition, so this is us super happy. So iGEM is a great, it's a great competition for people that are already within the scientific education. So these are biology students. But how do you communicate with people that are absolutely not involved in that? So I had the idea to create like a video game, an open source video game, because some of the principles of engineering synthetic systems are very simple to understand. So people are sometimes scared, they're like, oh my god. So it's rational design. So, so the idea was that the players could craft genetic devices to give some specific skill to the cell in the way of like Minecraft or something like that. And so what we really wanted to do, so I got some game designers and programmer folks to work with me, and I convinced them to do this, which is basically a real game. We wanted to do a real game. So there are many games that are successful for academia, like Foldit and Eterna, which engages the public, but they're not real games, they're not fun. So we wanted to, I'm sorry, but so we wanted to do something super engaging that the kids could play, and at the same time when they learn in the news what is synthetic biology, at least they have a little bit, or they are more familiar with it. So what I learned from this experience is that I was very young and naive, because I decided to do this in the academic environment, and it didn't go well with the scientists. They were like, what is that? I was like, it's a game. And so yeah, so you really need to you see the barrier between what the kids like and what the scientist likes. So this was a nice experience. Then I got involved with some designers and trying to explore how creative approaches could nourish at the same time scientists, but also have like a feedback loop, like learn between each other and how to communicate. So I designed some workshops with a friend of mine who's a designer, and so we were putting together biology students and design students to just like meet up and think about these issues and come up with a creative project. So this was about the body, so we call it Bodywear Workshop. So it's really to think about you as a biological entity within this technology, learn about your body, and also how you can engage with it. And then I got involved with the artists, and this kind of like changed my life, to be honest. If you look at the dictionary, art is human effort to imitate, supplement, alter, or counteract the work or nature. This is like is exactly the same as synthetic biology is supposed to be doing, right? So this is kind of like, it says something. And so why art? Why combining art and science? And I'm gonna quote a super artist scientist, Joe Davis, and he's right. This sentence says it all. Art helps science understand itself. And the reason why is because, yes, the artists are able to not only understand what we're doing, but also to even stretch the boundaries of what we're doing and really critically engage with some things that as a scientist in academia you would never do because, yes, you're formatted. And I was really amazed by the fact that artists can precede scientists in conceptual breakthroughs, and you learn how to do science differently. so yeah, so they offer glimpses to what is possible and what is contestable. So they really bring an amazing work into this. So I participated in research platform for art and synthetic biology called Making Life. This is by the Finnish Bioart Society, where we were basically doing that. So these were professional artists, and I was sad that I was the only scientist there. So if you want to work about something related to science, please invite us. We're not that scary. We can learn a lot from each other. And so, yeah, so we were providing insights on the connections between synthetic biology, technology, and society, and it was a lot of fun. And we came out with a project called Living Ashes. So the next session is going to be a workshop where we're going to do some of the practical things that this project was about, which is basically tackling the concept of the definition of life and also the ontology of artificial life, because yes, as synthetic biology is predicated upon the control of living things, and at the same time the concept of life itself is not very well defined. And so we were playing around with this, and I invite you to come afterwards. Just to conclude, so synthetic biology is really the only thing that you have to remember is basically applying engineering to the living systems, and we are in a potential future dominated by this mindset. And one way to encourage other frames of thought is by bringing the tools available to outside academia, basically, and serve other disciplines. So this also applies for any kind of technology, not just synthetic biology, but breakthroughs, cutting edge technology. So that's it.
We have 15 more minutes if you have any questions, or if you want to jump to the workshop, or if we have 15 minutes of break, as you like, or if you guys want to discuss.
Yeah, the next session starts in half an hour, so yeah.
Okay.
Yeah, the session starts 15:15.
So there's 15 minutes.
Okay.
So we have 15 minutes for questions, then 15 minutes break, and then workshop.
All right. Question?
Yeah,
There's
questions,
one
yeah.
question here.
Yeah.
Yes?
Okay. What your activities demonstrate once more, and it's a general issue, I would say,
Sure.
is that in universities are less and less places of knowledge, and especially less and less places where it's fun to go after knowledge.
Sure.
So how do you see this displacement, which is actually taking place consciously or not consciously, of real knowledge? I mean, useful knowledge, knowledge which helps people, which also makes them fun, from university where it used to be, and as far as I'm concerned should be, to places like you guys are running. And also, how do you value? Because you tend to say, and it's quite honest from you to say, well, we cannot do as well, or our equipment is not up to the standards, etc., etc.
Of course, yeah.
But I think slowly, your knowledge will be at that standard. So how do you see that future?
So I think it comes down to social organisation is the real difference, right? And that's why you're never going to have £1 million research efforts coming out of hackspaces, is because there's no one there to organise it. People do it for fun, and they therefore do their own project. No one's paying them, and so they want to do what they want to do. There's no one saying, well, I'll pay you to do something that you might find interesting, but is not directly what you want to do, right? There's that kind of payoff, I guess. And again, and I think also, firstly, at the moment, we definitely don't have the same capabilities as, all these massive pieces of equipment, because that takes a lot of infrastructure as well. And those infrastructure jobs, the jobs that people have to do to maintain that infrastructure aren't interesting necessarily, but you still need someone there to do it. They need to be paid to do it. And that's not really going to ever be the case for hackspaces, I don't think. But that doesn't mean it's not doing something interesting, because it's doing something else. It's not competing. It's kind of looking at maybe what you might call low-hanging fruit, right? So the universities, they might be kind of like, you know, these million-pound efforts to scale Everest sort of thing, to get to the top of whatever the next kind of big breakthrough technology is going to be. Well, hackspaces, they can look around in the hills that the universities haven't thought to look in. And it's not necessarily a hard trek, but it's one that no one's done before. So, you know, I think that's the kind of placement of it all. Yeah, so kind of low-hanging fruit, but stuff that hasn't been done yet.
Yep.
Can you please elaborate upon the process of obtaining the license?
Yeah. Yeah. Okay. So again, I can only talk about the UK, right? I don't know how it works in other European countries. What I can say is the UK legislation is based on EU directive, and so therefore it will resemble a whole bunch of other EU legislation in other EU countries, right? But so in the UK we have what's called the HSE, the Health and Safety Executive, and they're the ones in control of who is allowed to work with GM crops, GM organisms, whatever, or pathogens or things like that. And so essentially you have to notify them of your intent, and that basically means filling out a form and saying, look, these are our procedures, is this okay? And then you pay them £500 and they get back to you within 2 weeks. And in our case, they said yes first time, right? And so one of the important things, I guess, is before that, you need to have a limited company. Certainly in our case, you need to have a kind of legal entity that is able to deal with that. And a legal entity for biohackspaces is kind of necessary anyway, because you need to be able to deal with kind of chemical reagent suppliers, and they'll never really sell to individuals. But if you're a limited company, they'll happily sell to you. So you need one of those anyway. You know, I don't know, again, how it works in other countries, but certainly in the UK, it, you know, takes a day to set up, and it's pretty much free, a limited company. But then, yeah, the hard part was making sure all our documentation and all our operating procedures were kind of up to scratch. Yeah, and then it was a case of filling out some bureaucracy.
Were there any costs involved with? Is there any requirements for specific tools, for example, destroying organisms after?
Yeah,
like that?
yeah. So there is a. I mean, I could bring up a spreadsheet of all our kind of essential things we have that we need to keep in stock for the purposes of doing any CL1 work, any genetic organism work. So you need to have, you know, a disinfection procedure and things like that, and crucially a sterilization procedure. And what they really want is like an autoclave. And so I think if you're going to be doing GM stuff, it's kind of crucial to get that. Required, yeah. Whereas technically you should be able to use just a pressure cooker, but I don't think that's really going to be, you know, they're not going to be happy about that probably. Yeah.
There's two questions here. I mean, you on the back, yes.
Yeah. You said in the beginning that for biohacking you take a longer time to fail.
Yeah.
So how do you approach that problem?
Okay, so I think you have to. I think the best analogy for it is not like. I think a lot of people say, "Oh, it's like the early days of electronics and computing," right? I don't think it is at all. I think it's a terrible analogy. I think it's much so with you know programming and things like that. If you know nothing about programming, you can start very simple, and you can try and you can fail, and you can try and you can fail, and you can do that 50 times in half an hour, and eventually you get it. You can't do that with biology. It's much more complicated. You need to know much more before you even do the first thing, right?
And there's a lot of unknown stuff.
And there's a lot of unknown stuff that even you know any biologist like who's done a PhD or is a professor, they're not going to know that either. So you're contending with the unknown a little bit. So electronics, you know exactly how a chip works, right? You have a data sheet, and that says exactly what happens. There's no data sheet for E. coli. There's guesses, basically, and they're getting better and better, but it still doesn't tell you everything. And so I think and again, so a lot of planning goes into it, right? Because also it's more expensive than messing around with electronics. So it's within reach, so for £200, £300 you can do a really interesting project for yourself. Even cheaper than that, there are lots of non-synthetic biology projects you can do for much cheaper. and it's much more like maybe amateur rocketry, right? You plan and you plan and you design the whole system and you put it together and you set it off and it blows up and you're like, ah, well, and then you have to start again. And so there's a lot of planning and then a very kind of reasonably short execution time, and then you go back to planning again. So it's a different mindset. There's a lot less tactility to it. It's not like woodwork or metalwork or coding, where you can just trial and error. You trial and error, but you do that very slowly, so you have to minimize the error, if you see what I mean.
Okay, so you can't parallelize the process somehow.
So, I mean,
So there are some components that have been done. So there's like a registry of biological parts. The goal for that was that, basically, to be able to characterize biological components in the way that you would characterize an electronic device. But there's still a lot of contextual issues. It depends on many things, so you cannot really do that. But there's a GFPG will give you GFP. So there are some things that that you can do, like that if you know how to do it, it's very easy to do.
Yeah.
I wouldn't say that it's that difficult.
Yeah, exactly.
But there are a lot of then when you want to create something new and explore a known field, then is when it gets more complicated. But there are a bunch of stuff that is today well known, that anyone could do in my opinion.
Yeah. So yeah, there's lots of things that can be done, you know, without so much trial and error. But you have to accept that sometimes biology can be unpredictable. So.
There was another question here.
Yeah.
Site specific, but how does the hairy cactus actually work?
I don't know. I still really want to meet the person who did it. So
I think it is conceptual work.
no, it's not. It's real. It's real. So I know the.
Oh, are you sure?
Yeah. So Howard Bolan, who is the.
It's conceptual. Yeah. Yeah. I didn't.
Really? So Howard told me it was real.
Well,
I mean, it's
you would
real
do it
hair,
with the
but
artist
you
who made it, right?
So I need to. I've never met Laura.
Okay. So it's like a transplant. The transplant is some hair.
I think it is.
I
Yeah.
think
It's
conceptual.
It's conceptual. Yeah.
Howard lied to me. What a bastard.
Yeah.
I give up. This is all pointless now.
There is a window here. I mean, they made it as a sculpture, but I don't. As far as I know, it's not.
It's not great. Yeah.
Because I thought it was conceptual first, and then he said it wasn't. So I can make it for real, maybe. Yeah. Oh, there we go. That's another. That's a bit of a more of a mountain to climb. So
Other question?
Yeah.
We have 2-3 minutes.
If you consider starting up a bio party, if you're hackerspace, do you have any advice besides what?
I think just start with the people. Find out who's interested.
Community.
And regulation.
But I mean,
That's gonna be your main obstacle.
okay, no, I think there's so I focused on the genetic engineering part, partly because it's a nice headline to get everyone in the room. But there's a lot. But basically, there's a lot to say for starting with stuff that's got nothing to do with genetic engineering, right? There's a lot of interesting things you can do in terms of going around doing DNA barcoding to catalog all the species in your surrounding area, things like that. There is molecular biology. And will get you thinking about how the biology works before you even have to, all other bioart projects that maybe manipulating biology in non-genetic ways. There's plenty to be done there. Growing fungi in interesting ways, or mold, or things, all sorts of things. So yeah, you could even start, there. Don't necessarily begin with the idea of we must do genetic engineering now. Work your way to it. Because that's certainly how we kind of did things.
If you have any more questions or whatever online, just go on FSCONS Twitter. You can ask questions and we try to respond. We are a small team in Paris. Well, thank you very much.
Thank you.